Hello and welcome to this video on the idea that the DNA test you take to trace your ancestry could be weaponized. DNA test kits like 23andMe, Ancestry DNA, and more are all viable options for the average person to get a very general idea of their genome. They can be vaguely useful in tracing your background to some extent, identifying some commonalities in certain diseases you might be more prone to or carry the traits for. There are also some very tenuous claims that the knowledge contained in these tests could be used to make a biological weapon system targeted at just one person or group of persons. The gist of the conspiracy and very delusional idea is that the tests, or at a minimum, results are shipped internationally. These international tests or results are more than what is sent to the consumer. It is, in fact, true that the data extracted from DNA tests is far more than what you get back. With a bit of knowledge, you can extract a lot more information that you would normally have access to based on the files that are sent to you, but there's also much more that is not accessible in this data. Even if it is not available now, if the samples are stored correctly, further information can be extracted. This is the data that is supposed to be the source of biological weapon systems. It is in theory data about every bit of a person's genome, a complete sequence of every gene that makes them up as an individual, their blood type, bone marrow matching, ancestry, diseases they carry, and possibly exposed to, and more. A lot of information on an individual and possibly a collection of individuals. This is where the juxtaposition between gene therapy and biological weapons appears. It is only through the knowledge of exactly which genes and how those genes are mutated, missing, or otherwise dysfunctional that we can create treatments for genetically associated disorders. In the case of hemophilia, a gene is used to substitute for the missing or malfunctioning gene and therefore allows the production of missing clotting factors. COVID-19 vaccines allow the creation of spike proteins which stimulate the immune system and prime it for future exposure. These and other treatments are all currently available. The existence of such therapies is also why some less well-informed individuals believe that genetic information could be used to make targeted biological weapons with your DNA information. They think that if you know the genome of a person, you can target them. Unfortunately, in some ways, and certainly fortunately in others, this is not the case. We say fortunately and unfortunately, as anything that could be considered a targeted biological weapon in this case could also be used for mass treatment. Imagine if you had to administer an anthrax vaccine en masse, or to remove mosquitoes carrying an endemic disease. Well, it's actually true, the last has happened, but that example will also highlight the problem of this idea, and we'll discuss it later in this video. But keep in mind that on one side of the equation, we have been able, to some extent, use this as a weapon, but under very narrow circumstances. Under the other, it's completely impossible. Fortunately for some, even the egalitarian ideas we've just described are not possible. No matter how much you know about the genome of an individual, you cannot get a targeted weapon. Genes are nearly always universal. No one gene, or in fact several genes, is likely to be enough to target one person among hundreds, thousands, millions, or even billions. Even when we talk about forensic DNA testing, it is a probability based on a small number of mutations in a specific place. These mutations overlap with others who have the same mutation in one or more parts of their genome. By taking the total probability of the different mutations, you can narrow down the probability of one individual, but you cannot completely exclude everyone else. The other key to this is that you take many different, very far apart parts of the DNA. Put another way, look at this image. There are distinct sections. Each is made up of its own sequence. You want to target just this sequence here. The problem you face is the same sequence also shows up in these other 5 sections and correspond to genes in those parts of the DNA. On the scale of a whole human, this issue will show up many, many times more often than what we've shown you here. You cannot target one section of DNA very easily without targeting everything else at the same time. This is also why DNA tests cannot be used to target biological weapons to one person at present. You cannot customize any system to one individual. The obvious solution to many is, why not just make a bigger target site that is more specific? The more it has to match with, the less likely you are to get crossover with some random individual. In theory this would work, but in practice it does not. The idea has been tried, tested, and failed with PCR technology, and PCR technology is something done in the confines of a lab under optimal conditions. As any soldier will tell you, technology in the field will fail, and it will fail at the worst possible time. In this case, big genetic sequences just do not work with this sort of system or approach. You need a small primer for PCR that can fit to the gene sequence or the area that initiates it so it can be read. Bigger primers do not work. There is a rough upper limit for primers of 200 base pairs. Most are well below this in the 20 base pair range. If we take this limit to be true of a weapon, then it would be similar to what occurs with the COVID-19 vaccine, amongst others. The vaccine uses RNA, but the design does not use the whole 1,200 to 1,400 base pair long sequence, but a very small subsection. This can be between 20 and 120 base pairs. This only creates the area relevant to the immunological response. That is why you can use a smaller sequence and why it works. If you make it bigger, it does not work and could not be used effectively. This is why RNA, or in fact DNA-based COVID-19 vaccines, work to protect the body, and what makes them work. The challenge for you is, how can the opposite result be achieved? Not adding function or genetic code, but disabling it or targeting it. This is also where the aforementioned challenge is. How do you target a single person among millions or even billions? There are no singular genes unique to an individual in just about every case. It is also incredibly hard, if not nigh impossible, to specifically target one person's particular gene, let alone genome. That means anything developed is going to be affecting a lot of people, not just who you want to affect. The only viable means of targeting someone with a biological weapon derived from genetic data is not based on genetic data at all. It is to use a process like the inclusion of clotting factors, but to use a much more nefarious gene. For example, the inclusion of a misfolded protein. Such a situation would lead to issues to do with the functionality of anything that protein is involved with. This has nothing to do with knowing the genetic sequence of someone, and so it is just as non-specific. This is true of any receptor, gene, biological process, or other information that could be gleaned from a DNA test. There is no new information from it, but just specific information on which version of a gene, receptor, or anything else on a specific individual. The final hurdle with this is administration or perhaps infection. How do you get to the targeted individual? In the case of mosquitoes, they were specifically bred in a laboratory, and they were bred so that they were sterile. That is how that could be done. By releasing them into the wild, they then crossbred, and anything they crossbred with did not produce offspring. This ensured that mosquitoes were unable to continue to cause infections and disease. However, that was the end of the line, and you had to have very specialized conditions for it to even begin. Deploying a weapon on the battlefield or in a civilian environment on the basis of getting one person is nowhere near as simple as what you would have in a lab. In simple terms, no, you cannot create a targeted biological weapon by weaponizing a DNA test. What you can do is get a lot of information on the person, who they are, where they came from, and what they've been exposed to some degree. At present, the safest and most effective method is a tried and true assassination technique. Good old-fashioned radiation to mutate the genome of the targeted individual. This is not to say things may not change in the future. Since the Human Genome Project was first finished in 2003, we've been able to grow the technology at an amazing pace, and even now make whole genomes synthetically with completely made-up organisms that we've customized to a particular purpose. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below,